now is a good time to talk a little bit about the clinical trial data, and then we can maybe have a discussion about the toxicities and the management of the toxicities. Obviously, one of the toxicities, as you mentioned, is cytokine release syndrome, which requires that hospitalization. I think it's important to note that the clinical trial that led to the approval of this agent was built upon phase 1 and 2 testing. The initial trial of this agent was actually in cutaneous melanoma in patients who'd had surgical resection. It was presented by one of our colleagues, Mark Middleton, at an AACR meeting about 5 or 6 years ago. And I remember sitting there and watching, him give this talk and thinking, wow, this is a really cool, neat thing, but that's a weird population to start, looking at a drug like this, and it sort of fit in with the field of melanoma, where there was a lot of adjuvant studies of vaccines, and this was sort of like a weird kind of vaccine, maybe. Obviously, it's not that. It's a very different type of molecule. But then soon after, it transitioned to being looked at in both cutaneous and uveal melanoma. And I think, to your point, the expression of this GP100 peptide is much more consistently expressed in uveal melanoma than cutaneous melanoma. And for reasons that probably are immunologic, cutaneous melanoma is so genetically altered, typically by UV irradiation, that it has to come up with all kinds of mechanisms to get around the immune system, whereas uveal melanoma is much less genetically disturbed. And so, it probably is just better able to remain differentiated, as opposed to the dedifferentiated subtypes we see in cutaneous melanoma. And so with that expression of the protein, it just seemed like patients with uveal melanoma were doing better. But it's interesting, doing better doesn't mean high response rates. We've treated collectively a number of these patients, Dr. Hamid, and I have a few responders. You probably have a few responders. But we have a lot of patients that have been treated for a while on this drug, didn't respond, but survived longer than we might have predicted. And in fact, that's what was seen in those early studies, so that patients just seem to be alive longer than we thought they would be based on their disease. And so based on that, the design of the study wasn't looking at response rate, wasn't looking at progression-free survival, which are typical metrics for primary endpoints on definitive trials, but in fact the primary endpoint was overall survival, period. And indeed, it met its overall survival landmark, or not landmark endpoint, but comparison of the Kaplan-Meier curves. And in fact, the overall survival hazard ratio for overall survival was 0.51, which is about as good as you're going to find in a clinical trial of a drug. Now, the way the trial was designed, it was 2-to-1 randomization. Patients were either randomized to Tebentafusp or they're randomized to an investigator choice. We had 3 options in the investigator choice. We could use single agent pembrolizumab, single agent ipilimumab, or dacarbazine. Most people recommended pembrolizumab as the investigator choice. Second to that was ipilimumab, and third to that was dacarbazine. And ultimately, this was a positive trial that has led to the FDA approval of this agent.